So it's great to have Sagi Snir here. Sagi is a professor of computational evolution at University of Haifa, and he's president of the Israeli Society for Bioinformatics and Computational Biology. He got his PhD in computer science from the Technion, I guess at the same time as Julia, around that time, so they were there together. Focusing on analytical, algebraic, and maximum likelihood solutions to phylogenetics. He was in a postdoc in math and computer science departments at UC Berkeley, and then joined University of Haifa, and he there established the bioinformatics program. And so it's great to have you here. Today he's going to talk about, you'll tell us.
So it's an honor for me, and eventually the opportunity that I went for so long to come and meet eventually Julia, and she's not here. So but also Nati. So that we all share some previous origin, but and it is the title is Harnessing Genome Dynamics for Phylogenetic Reconstruction, and it is dedicated to my advisor Ben Shaw that passed away tragically 3 years ago, and I still miss him a lot. Anyway, so let's start. So and this is another something. So the Israeli Academy is committed to humanity and democracy, but we still at the same time must remember about these 130 hostages that no one knows where they are. They were kidnapped from their homes, and they are dying and tortured and raped every day by the Hamas. So let's start with. So what is comparative genomics? And actually, my talk here will be maybe in a way to convey or to sort of introduce maybe something about biology. So in comparative genomics, we take a genome, a complete genome of some organism that alone does not tell us too much. But when we do that over several species, several genomes of model organisms like the E. coli or the fruit fly or the guys that you see here, plants, we can learn something about what are the differences and what their importance. So this is something nice that I found out in the literature, that actually evolution that we learn so much is actually only a tool in comparative genomics by leaving these footprints that we trace and try to understand what they really, these footprints, these differences, how they really affect. And actually my talk will be dedicated to this. And just to tell you that I prepared, that I worked hard how to convey here to, because this is the audience I like to speak to more than like real biologists, people that understand models, understand in particular and stochastic models. Okay, and let me, this introductory slide, let me cite something that every day I agree with. Nothing in biology makes sense except in the light of evolution by Dobzhansky. That was said, I think, in 1973, something like that. Okay, so what is the talk outline? So I'll introduce the jump model. Then under the jump model, we'll see how a genome or genome evolving is a continuous time Markov process. Using that model and that Markov process, we will reconstruct the prokaryotic phylogeny using the synteny index. Wait to see what it is. I'll show results on real microbial data that, in a way, I'm excited to because here we will forget about biology, but we will return to biology later on here. Then again, we will forget about biology and return to mathematics. Let's see if we get into here. So normally evolution is perceived as a tree-like process with ancestor and descendant and a parent and a child, and this gives rise to that what we call a tree-like evolution. And well, he was not maybe the first one, but now we associate that to him, Charles Darwin, in the 19th century. So descent with modification, this is the term that he coined. And what is actually a phylogeny? A phylogeny represents evolutionary histories. The leaves, we put at the leaves extant species, living species. And internal lodes represent either extinct species or simply speciation events, as these represent evolution. Okay? And branches represent evolutionary relationship. Now, let's be more precise. So standard phylogenetics, that art of reconstructing this tree. So we collect, this is called multiple sequence alignment, and we collect the same gene, or we call it orthologs, from different species, and we compare them. We compare. And now we look for a tree that accounts for all these differences that we see here. Okay, the differences that we see in the multiple sequence alignment. Okay, columns that somehow the footprints of evolving along that tree. Okay? Now, if we have a method that converts these differences to the true tree, now I start with mathematics. So we assume that was evolved along some true tree, and if we have a method that converts these differences to the true tree, the method is called consistent, which is very conventional and standard term in statistics. Okay, statistical consistency. Okay, now when we are giving the true distances between the leaves, okay, the distances along the tree, okay, in the form of a matrix. Okay, the distances along the tree, we know actually, and I will return to that along my talk, we know how to construct that true tree. And this is important to remember, and it is important to say now. This is why I inserted that, because it used to be a problem of inference. I mean, my problem. Okay, so the conclusion. If we have that construction or black box that constructs from true distances the true tree, we only need to correctly infer these distances. This matrix needs to be correct to hold these distances on the tree. Okay.
Technical. So consistency usually refers to a limit of infinite data.
Yeah. Sorry, I didn't say that. Exactly.
Are we talking about a limit of an infinite number of species?
Oh, that's a great question.
Okay.
Okay. I will return to this a lot. This is the talk. What about this consistency? But this is the amount of data.
Size of the genome.
Exactly. Samples. These are samples from what we call the true tree. Okay? So it's not about. So this will be infinite or not large enough. And this is always this is n. Maybe my talk n. Yeah. Okay, so we need to correct the infer, and this is actually what the crux of the problem, how to really correct the infer, and everything here is about modeling. Okay, so but it is tied into the biology. So in the 80s, Charles Wars found the archaeal domain, which are some unicellular organisms. But then this is important. Then he found that gene that was ubiquitous and fundamental and very preserved among all these guys, that he could actually take that alignment and construct the tree of life. A phylogeny stretching back to dawn of life was constructed again by this guy, Charles Wars. Okay, however, that was at the beginning of that decade, but at the end of the decade, in Research News of Science appeared that title, Genome Data Shake Tree of Life. New genome sequences are mystifying evolutionary biologists by revealing unexpected connection between microbes, so to have diverged hundreds of millions of years ago. And these are these connections, the unexpected connections, so these are the chloroplasts that we find in plants. This is the mitochondria that we find in all of us, but they were transferred somehow very early in life, in history of life. However, so classical evolutionary school, the Darwinian school, results in a tree-like relationship because we have that descendants. However, some very significant biological processes result in a network relationship. Let's see what is this relationship. One such process is lateral or horizontal gene transfer, which is the transfer of the genetic material between distantly related organisms. Okay? And these are these unexpected connections. Now, lateral gene transfer is mediated by three processes, mechanisms in that I myself am not very expert. One is viruses. I think this is the viruses. These are plasmid, and these may be free DNA that moves, travels in the air, in the open air. However, this is reflected by horizontal edge that we see here on that tree, meaning that that gene was not inherited by the parent, rather was transferred horizontally from someone else. Okay. Now, HGT or horizontal gene transfer is very common among microbes. If we take here these three strains, only three strains, very related organisms of E. coli, they share in common only 40%, only 40% of the core genes they share. And it is, so this is how rapid it is, and this is a very significant mechanisms in their evolution. This is why they evolve so fast, and they develop toxicity, and everything is so, or develop antimicrobial resistance, antibiotic resistance by HGT. However, it has become quite an agreement that even prokaryotic evolution adheres in large a tree shape, because if the mass of the genes really inherited vertically, most of our genes as microbes are inherited from our parents, then the main thread of evolution is, after all, vertical. Like we see here, the core of the genes are inherited, but we see all the time like these small connections that represent gene transfer, vertical gene transfer. Okay, now we come to the big questions that I tried to answer in the talk. So HGT actually hampers phylogenetics, and we've been here. I mean, we started with this as quite young researchers during my postdocs, and it was all the time in the air that HGT actually, this gene transfer actually hampers, because if you try to align these genes, you will get a wrong tree, okay? You will get a wrong tree because that gene was not inherited from its parent, rather from someone else, so you will get a wrong tree. So it hampers phylogenetics. However, can we actually use that HGT? This is somehow inherently contradictory. Can we actually use HGT for phylogenetics? And next, why should we do that? Okay, why should we use HGT for phylogenetics? And I will try to answer them along the talk. And please don't be shy to, if you find something interesting or unclear, you are so welcome to stop me. Okay, and can we use that signal for other purposes? Okay, and since it becomes now so clear that HGT is so crucial and so rampant, so we all the time we find more and more uses for that signal of the HGT. Okay, so in 2014 we started by some like Satish, my advisor Satish, I used to say some hack. And the hack was, in my group, I started with defining that thing called the synteny index, and let's see what it is. So the 2K neighborhood of a gene, let's look at this gene G sub 0, okay, in some genome, capital G1, say. And the 2K neighborhood of G sub 0 is actually the guys that are K genes to the right and K genes to the left. And it's important here to note, normally you hear a lot about DNA. Here the resolution, the level of resolution is only genes, okay? So of course every such guy is some sequence of nucleotides of DNA, but we collapse that into just a sequence of genes, okay? So and we speak about the 2K, so we have the 2K neighborhood of this gene is the K to the right, K to the left, and this is the set of, or the 2K neighborhood of that gene. Okay? Now, the synteny index of this gene G between the two genomes G1 and capital G sub 2 is the proportion of shared genes in these two neighborhoods. Okay? The Jacquard index, what is called. Okay? So it is what is written here. Let's be more vivid. So for example, for k equals 3, like we have here, okay, 3 here, 3 here, the synteny index is first E is shared, then I is shared, and now F is shared. So we have 3, and if we normalize by 2k to be always between 0 and 1, so we get that half of the genes are shared in the 2k neighborhood of G, of little g. Okay, and we have also for X that is not shared, we just a convention, this is 0. Okay, now if we average that synteny index, not only on G, but average over all genes that are shared between capital GI and capital GJ, okay? So that average, this is the average. So for every gene in the shared pool of genes, we take that SI, okay? And we also here normalize. So that average will always be between 0 and 1. So we normalize by, say, 2n, where n is the, or by n, where n is the number of times that we sum here in this sum, okay? And this thing about, and the reason we did that back then, 10 years ago, is to combine both gene content, the proportion of genes that are shared, but also the order, how the order, and you will see later on how order is reflected here, okay? So it combines both the order and the content. Okay. So this average, bar SI of K defines some similarity between the genomes.
Can you go back? It looks like you're thinking of it as between 0 and 1. I'm wondering, can you go back? Can you go back one slide?
Yeah.
Is this supposed to be an average rather than a sum?
No, you're right. That term I did not say. Yeah, that term is missing. But that term of 1 over n is missing.
1 over
The size of capital lambda.
1 over size of lambda.
Yeah.
So is it 1 over the size of lambda, or 1 over the size of the genome, or what are you normalizing?
You're
like
right.
The total number of genes, or just
normally, I think it's not lambda. It is n because lambda ignores the guys that do not exist. So you want to account for.
So n is the number of the
the
size of G1.
size of the genome, maybe.
The size of G1 or G2.
Okay.
So
Yeah. So this term is missing. Yeah. I should add it. But I say it all the time. Okay. But this is a measure of similarity. Now you remember this term. This is this matrix of this similarity score. You remember that matrix. It is between every two genomes in our set. Okay? And now you can give it to this algorithm that constructs the true tree. But this is not true, okay? But still, you can give it, okay? But so we are not guaranteed in anything. We actually convert the SI. 1 minus SI give us some dissimilarity score, and this is some heuristic. We have a dissimilarity distance matrix. You can apply standard cherry picking, which is exactly that algorithm that gives us the correct model if this is true. And it is called neighbor joining. It's quite a famous algorithm for this dissimilarity matrices. Okay. So the above SI and its reconstruction-based approach is actually a heuristic. We could not say anything about it. It's simply a heuristic. And modern systematics and evolution in general, or genomics in general, nowadays it becomes more and more prevalent and common to use rigorous models based on which inference can be drawn. So at least for me, it is important these models, but also to other biologists. Like I said, I'm in this business of gene transfer for quite a few years since Maui. after my postdoc, so it's like 20 years now. And we were modeling HGT through several approaches, tree branches or gene content. But however, I don't remember I encountered before, to the best of our knowledge, not as a driver for loss of synteny or loss of gene order conservation. So gene order conservation, this is what all this inference is based on. Okay, so then that's why we came with this very simple, very abstract model that we call the jump, and we say let's see what we can do with that. So let's consider the following model: a genome G with large. This is the large here. This is different, large N, and we will say why it is important. Now every gene G by a Poisson process with rate lambda jumps to another one of the other n-1 slots, okay? And the target position is chosen uniformly at random. So you have here very simple but well described and well defined model of evolution, and let's see what can we do with this. So this is the jump, okay? So and you have that process. So if we speak sometimes about mutation process, so this is the same process, a Poissonian process of every gene once in a while jumps to some other place, and this is also important why that N the genome needs to be very large. You cannot say infinitely large, but very large. Okay, so now why we use that jump model? So the idea is that the jump model corresponds to several evolutionary processes such as translocation, gene gain, and loss, and we can explain these processes somehow by the jump. But the other importance or advantage is that its simplicity allows rigorous analysis that I was very surprised that we could say something, and apparently we can. So yeah, and this is why it had to be so simple. Okay, so the assumption of large genome allows asymptotic results, as will be shown. Okay, so you will see exactly. We need somehow to neglect several factors, but if the genome is not big enough, we could not say that this is a little O of 1, okay, something that we can ignore. Okay, so the above modeling defines a genome as a continuous time Markov chain, because continuous time we have these jumps that occurs in long time, but then it is a Markov chain because we travel in this state space of genome, because every jump brings us to another state in that state space. Yes.
So this looks like a transposon model more than a horizontal transfer.
Yeah, of course. But I don't know so much, but the reason I came is exactly because I analyzed that SI, that synteny index, and I said, let's devise something very simple, and really, because I think transposons are in eukaryotes, but also in prokaryotes, but in I don't know if people speak about transposons so much in prokaryotes, but I really said, let's define something, I mean, even irrespective of biology, let's define some process that may, but still you can say that a jump of F to here represents some swap or translocation, or maybe a gene loss here and a gene gain here. This was my intuition. Something that I will be able to say about the SI, because we used the SI and it became like expected from us to say something statistical on it. But when people from eukaryotes speak to me about the jump, they say, "Hey, we can do that also." And I say, "Yeah, welcome. Let's discuss that." So, okay, transition and their probabilities represent the jump. So, like I said, the jump here. So this is the state space, and every state here is a genome. Is some. Or nowadays we discuss a genome as a permutation of genes, okay, that we have here, the permutation of some permutation. So this is the space of permutation, and you jump from one state to another, or you move by a jump, okay, and this is, of course, a stochastic process because of the definition of the jump. So this was the higher level of the state space of the genome that we move here, and we will see. Again, we are receiving two genomes that were somewhere that in that state space, and we want to find how many moves, how many jumps were or led to from here to here, but of course in a more probabilistic way. Let's dive down to, because we are after that synteny index, let's dive down and to analyze at the neighborhood level. So this is the lower level, the gene neighbor, as actually a different stochastic model, which we call dispersed immigration process. So see this. So we model gene neighborhood as a dispersed immigration process where genes jump into and out to. So we have a rate of lambda of insertions at every such position. Why lambda? Because everyone chooses this position in probability 1 over lambda, but we have lambda guys that jump here. So this is lambda. And we have that lambda of this guy jumping out. So jumping in and out of the neighborhood and affecting this composition. Okay? So, and let's see something here, something interesting. So imagine this horizontal G sub A is the horizontal gene that jumps here. So actually it pushes out from the neighborhood that G sub L prime. So L prime is not anymore in that neighborhood, okay? And imagine conversely that some gene jumps out, for instance, this guy, which not necessarily that same guy. When this guy jumps out, that guy either returns or enters that neighborhood. So this is how the neighborhood behaves. So this is how the jumps in and out affect that neighborhood. And we denoted that xt as the distance between two guys, the reference gene G sub L, and some other parameter gene G sub L prime. Okay, and xt is the distance, the number of genes separating between them in the genome. And we want to know how that affects the membership in the neighborhood. Of course, in terms of time and that rate lambda. Okay? Because this is our SR. So that distance xt is affected, like I said, the number of gene jumps in and out from that interval. Okay, so it can be pushed away or it can shrink, get closer. Okay, and over long periods we have more jumps into and because we have one more slot than the number of genes, and this is why they get far apart. Genes get far apart as long as they did not jump themselves. Okay. Okay. So so here is that gene. Wait. So so now this is quite standard in stochastic processes. Pij of t denote the probability that xt to be at state j, given that at time 0 it was at state I. And with this Pij of t, we can actually analyze all these distances or the movement of the genes, and will be helpful or will be necessary for us to say all these rigorous things on SI. Okay. So XT can be described as a birth-death immigration process affected by the rate lambda and t. Let's see why. So we have that here. So this is XT, and it represents exactly the distance between, remember, G sub L, the reference gene, and G sub L prime. So this is the distance. And we have that rate of genes entering that interval. This is the interval. This is the distance between these two genes is now 3. So we have that rate of genes coming in, which is 3 lambda, and that rate of genes coming out. This is only 2 genes that separate. So we have 2 lambda. This is what made that special. It is not very standard death-birth process, which at that time we thought that will make things complicated and maybe necessitated our own intervention and development, because this is not standard birth-death processes. And this is why we had to introduce the immigration process. So at that time we could not, we thought that will make things complicated. The asymmetry here. Normally the rate of leaving that state is actually the rate of living here, okay, or the entering. Yeah, I need something here, but okay, so that equals to this, okay.
So it seems like there's sort of two things that are driving things apart. One is just what you said, that given where you are, the probability of going left is slightly less than the probability of going right. And then it seems like there's another thing, which is that the farther you are away, the faster you're moving.
Yeah,
right?
exactly.
So that aspect is also kind of driving things.
No, but this is birth-death. Birth-death, I mean, the more your population, everyone dies more. I mean, not everyone. Everyone dies at the same rate, but more death cases, and more birth cases. So this is exactly here. But I refer to that asymmetry here, that is exactly one more slot to enter than genes that jump out. Because when a gene jump out, then you move here, the distance decreases. So this is why we needed the immigration. It made us complicated. I mean, we can see how exactly we can derive, which is quite standard. But we didn't know all this stuff. Okay, and here, in some hand waving, because I don't want to delve here to these details exactly, but this is the theorem that so assume you have that system, okay? That system simply with lambda. And let G sub t, the genome after time t. And G sub t is, of course, very large. So this is the theorem. So you measure that SI that I described. This, for me, was really big surprise that actually we can from that SI that we measure actually derive that, which is the distance, which is that distance we are after. But let's see what is the obstacle here. So Q sub K of T is actually some average probability of these two guys that I described, but it is averaged over all members in that neighborhood to stay in that neighborhood. So it's some term that I found it really attractive or cute, may I say. But so this average probability that says something on this average SI, and this is quite not standard and also expected or easy, which is exactly the probability of these two guys not to jump. Okay, because once they jump, they are of course away. They are not counted. So this is why we need that large N. Okay, once they jumped, of course they will not jump to the neighborhood. Okay, so that is the theorem. For me, it was really nice. However, since we did, so this is of course some expression over pi, that probability to stay in that neighborhood. And because, as we could not solve that expression because we were not aware we were not aware, but we saw that we have here something that is non-standard, then we had to come with some heuristic, and which is very common in comp bio, to come with heuristics. When I entered that field, I was told that this is the rule, this is the reality, just heuristics. And so one heuristic was exactly that, -1 minus SI, and this is some other heuristic that is based on exponentially decaying system. We could prove something nice here, but we could prove something that this decays if we have that model, but it is definitely not something precise. Now,
but the way you use these things, you said, is just by greedily building a spanning tree or something, or a single linkage. The way you use these is just by then single linkage or something like that. Or how do you use these? I mean, these are numbers, right? You say you have two heuristics here.
Oh yeah, the heuristics. Yeah.
How do you use? I mean, this just gives you some distance measure.
Yeah, distance, and you give it to some.
But are these two? I'm just a bit confused because these two distances, are they monotone in each other, or because if not, I'm trying to understand in what way they're not.
Really
If they are monotone in each other, then does it make any difference?
We didn't care. As a heuristic, we don't
But
care.
if they're monotone in each other and the way they're using them, or does it make any difference which you use?
Which one you use between these two heuristics?
If they're monotone in each other.
What do you mean monotone in each other?
That both of them depend on this. That if one increases, the other also increase.
We didn't care because these are heuristics, so we never tried to prove something about this. We're talking about something more. I mean, along.
Missing something here.
This is what I'm going to talk about. Okay? So I just give that, and we will return to that in terms of biology. Because you can compute that. You can compute that. This we could not compute. We didn't know at that time what is QK.
So just to
Yeah.
So you're computing a distance matrix between genomes, and ultimately what you want is a phylogenetic tree.
Yeah.
But you're assuming some black box that's going to take that matrix to the tree.
Yeah, exactly.
So it's not obvious that monotonicity is going to give you the same
Well,
tree.
I don't know about monotonicity is really important
Is
here.
heuristic
I will
used in
return
this?
to this. But on
It's a single
heuristics,
linkage.
I don't try to prove monotonicity on heuristics because maybe I miss something, Nati.
No, but I mean, and this is ultimately both of them are. I guess maybe in this expression, what's measured here in the right hand side here? Which quantity? I'm a bit confused as to
What is this?
what is the quantity of all these?
Um,
What do you? I
you
mean,
are
this
right
is
that
you
it
take
should-
two genomes and I measure something between them. What is measured
So
here?
this
Because
is some normalization,
Right.
of course, and this is some heuristic that you can see that maybe you're right that this
No, but what?
supposedly this is monotonic. Supposedly this is monotonic. So you will for smaller SI will get more distant guys. Okay? And again to that that black box, the only thing I know to say that if it gets the correct distances, it gives the correct tree. So I cannot say this is correct and this even less. And we will even see that in, which is not important, but which I would say, but that I cannot account for. But even in biology, we will see that these two guys, the trees, are actually very distorted. So, but yeah.
So when you say correct,
What?
when you say correct, what does it mean for that matrix to be correct? Are we looking for the distance, the time of the least common ancestor? Is that what it's supposed to be representing?
So correct is number of events. Number of events separating the two leaves.
I see. In the tree.
Exactly. Lambda t. Okay, so recall that quantity we are after, lambda t, reflecting the expected number of events occurred along the path. This is in number of mutations, this is in phylogenetics, and not only in phylogenetics, this is what we are after in systematics. In the expression, this expression used to extract lambda t, we still have that unresolved component q sub k of t. And then, so we later on in that paper, we found our handmade or manually we were able, using generating functions, we were able to find that qk, but then actually we were commented that using techniques, standard techniques, that actually we could actually use standard techniques from spectral theory and orthogonal polynomials by Sam Carlin and McGregor. I mean, this is theory of at least 50 years back to solve these birth-death processes, and then we came. So once we found this Pij of t, we could of course plug it into this qk, this average qk, and found that. And here, Nati, this is where we were able to prove monotonicity. So this is q. Q is, you see, so lambda is not here because lambda is assumed to be 1, which is okay, and then your t is not only the time, rather the number of events. Okay, and this is just some example how q sub 2 looks like. This is some expression. Of course, all these are solved by numerical solution, but at least for us, this is analytical. It's not a heuristic. It is analytical. And so we use that here, but we still have that our own handmade solution, which is more combinatorial, generating function. But what is additivity? So a measure is called additive if we can translate its values along branches of the tree such that it correlates to the number of events. So we have, for every edge, we know how many events occurred on that edge, and this is additivity, or it is additive, and we can infer the number of events along the entire path. Okay, so recall that qk of t is the probability of a gene initially at the neighborhood to remain there, and this is some function over Pij of t. Now, why monotonicity is important here, really important, because we want to know that Si maps to a unique number of t, or lambda t, okay? And we needed, so of course, this is decreasing, this is monotonically decreasing, but this we could not, while we did not know that, we could not know. But showing that qkt is monotonically decreasing with t, we are done. Okay, so we see that 5 minutes, wow. Okay, let me rush. I mean, yeah, I delve into too much. Yeah, okay. Yeah, actually, let me rush over all this. Okay, this answers some questions. So, forget about, so this is the 16S. Remember 16S, that conserved ubiquitous gene. That tree is, so 16S is so conserved that you have very low signal. While our Si tree is much more expressed, which actually is quite known that this, and this is real data, that these jumps, these genome dynamics event, GDEs, are very incessant, like occurring all the time, in compare or in contrast to these mutations that are so conserved. So this is why we should harness the synteny or the loss of synteny into phylogenetics. Let me rush. And these are the trees. This is some NCBI tree that is not actually a tree. We use here some techniques that we defined long ago for other purposes. It's called convex recoloring. So the coloring here is convex. Every subtree has its own color, and now we see how convexity, how coloring remain not mixed under another tree. So this how this tree is similar to that tree. So this is the 1 minus Si. You see that distances here are very distorted, very much. These very long, and all the tree is very like clock-like. And this is this is the other heuristic. Some branches are extremely long, like thousands of events. And this is so I was really surprised with this, but again, it's nice outcome that, and I still think it is luck, but we can really rely on that exact rational function that we developed to infer correct events, and we use it further on with other projects that we run. So the conclusion here, the genome dynamic basis trees in general agree with the NCBI. Some significant incongruences found between NCBI and the exact tree exhibits realistic branch lengths representing expected number of genome dynamic events. Okay, let's keep this. So I have two. Okay, so I want to I want to briefly describe that the project that I run with my colleagues at the NIH, and these guys are big experts in prokaryotic evolution, and they among the guys that found the CRISPR. So, and they associate a big importance to being analytical, and they look and we look at Centenary blocks. And again, here let me describe very briefly. So again, we are under this jump process, and we ask ourselves how likely we are to see blocks, Centenary blocks of length 3, 8, under that time of separation. So this is some example. So this is edge lengths. This is how likely you are to see the Centenary blocks distribution. We are talking about Centenary blocks, so island distribution between two genomes. So here we have a relatively low jump rate, and we see many long blocks. Whereas here, under twice that rate, we see most of the blocks are very short, because the genome is sliced much more frequently, and then most of and so we had also some combinatorics here of really defining this precise distribution in terms of that lambda t, which is something nice, and that allows us to infer quite a lot about genome dynamics or expected genome dynamics, and we called it the neutral model of genomic islands or Centenary blocks. And I want to finish here. I have time okay, so genome dynamics events provide finer footprints than conventional phylogenetic markers, and you see that between these two trees. One was very lowly resolved, one was quite highly resolved. Harnessing these dynamics is essential to leverage these footprints into systematics, so we needed to come with something analytical. This can be done by accurate statistical modeling of these processes and devise tractable approaches. Results on real data should be confronted with other approaches. Now, we can say whatever we want, but we need to see that we really that this agrees with other knowledge of these processes, these organisms, these findings in other. And let me just, Mike Steele from New Zealand helped on all the statistical aspects. Haggai from Israel, Doron Zilberger, Doron is a combinatorician from Rutgers, and Christoph is some colleague of him, so they helped us about these tough expressions, a polynomial expression, a rational function that we needed some identities to infer. Gur, Danny Doer from Bielefeld, Yael were students, and Hepper and Jens Stoy from Bielefeld. CGSI support by Eliezer Eskin from UCLA. These are the guys on the SI, and these are my colleagues from the NIH, Eugene Kunin and Yuri Wolf, and again Mike Steele on the genomic islands. And that's it. I'm done.
So let's see. Everyone here is invited to lunch downstairs, so we can ask Sagi more questions while we eat. But in addition, if anyone would like to ask a question now while the slides are up, we can take a question now, or anyone has any?
Actually, quick question, just 'cause, so for eukaryotes, I mean, when you talk about like the genes appearing in order, at least for eukaryotes, that's not quite the case because they're all like mixed together. Is for prokaryotes, how much can you like, talk about just genes as contiguous parts of the genome, like that non-overlap? Ignore the issue of overlap between genes.
Yeah, modeling there for eukaryotes, correct?
So for eukaryotes, I know that you cannot do this. I mean, the genes are all mixed together. The exons are all mixed together. So for prokaryotes, I don't know if it's
you don't have junk DNA in prokaryotes almost, and you can do
the
that.
exons are. I mean,
There's no introns or exons.
oh, there's no introns and exons. There's no splicing at all in prokaryotes. Okay.
Oh no, yeah.
Okay, sorry. So that
Oh,
was,
no splicing. And you can really
Okay,
I
that answers.
No, I skipped these slides, but also inferring, like everything here is like combinatorics. But come on, are these gene D here and gene D here are the same? I mean, this is big deal of inference. This is what we call orthologs. So again, informally, I say I started with the combinatorics here that I found attractive, but it appeared to work. It appeared to work. It's nice that we came with that jump that explained, but again, back to irreality is. Genes do occur, so how you compute SI when you have multiple occurrences of the same gene even in prokaryotes. So we really need to first to model that and to, but I agree that also in eukaryotes it will be challenging, yeah.
So like kind of on what you were talking about, like in prokaryotes, it's a simpler genome, but a lot of the functional genes are duplicated, right? Like
exactly.
But is it kind of for your models, is it safe to assume that these functional genes shouldn't be jumping because they're kind of necessary? And if so, have you thought about like maybe how to like leverage that?
Yeah. So now again, you run into reality, and we all the time. We all the time, I wouldn't say criticize people, the way people like that rigorous analysis or rigorous attempt that we do. But they say, so for these genomes that we show the trees, they say, hey, come on, they don't have any genes in common except, that 16S. So there is no, the SI is fictitious. Now it's not because and still it works. So why it works? Because signal from other places. So many things here are in a way not realistic, like the duplication, like the copies, and still I don't know. People and us find it relevant and attractive, and because it also gives you it's a different approach, different measure of gene loss, and or the gene or loss of synteny, and we all the time discover more and more uses on this. So. But specifically, you must simplify the most you can in order to say something. I would say something rigorous or something. Something rigorous. You must simplify. We now specialize. It's not the jump. It will be insertions and deletions, and so we discuss all these things, but we don't know how far we will be able to model and stay with this monotonicity or these expressions that are tractable. I'm not sure on it.